Good morning, students. We are going to start lesson number 10, Human Health and Disease. In this lesson, in this video, that is lecture 1, we are going to cover vaccination, structure of antibody and human blood groups. Vaccination. It is defined as vaccination. It is defined as an administration of vaccine or inactivated pathogen. Now, pathogens are nothing but disease-causing microorganisms. So, those pathogens are partially activated or they are inactivated to protect against pathogens. For example, BCG vaccine, polio vaccine, tetanus vaccine, etc. Vaccine may be in the form of a protein or sugar from pathogen, a dead or inactivated form of pathogen, a toxoid containing toxin produced by pathogen, a weakened pathogen. So these are different forms of the pathogen, forms of the vaccine in which vaccine can be injected or it can be orally given, orally administered into human body. Vaccines are safe. Yes, vaccinations are safe. Vaccines are tested and go through many rounds of study, examinations and research. Now recently we have taken COVID vaccines. So you know that many trials are taken on human as well as on animals, and then only it is it can be published. So here is a chart of vaccination. You can just go through BCG, hepatitis B, then polio vaccine, then diphtheria vaccine, human papilloma virus vaccine, rotavirus vaccine, MMR, that is booster vaccine, typhoid vaccine. So many vaccines are given right from the birth up to 12 months. And then after the 12 months also some other vaccines are given. Let us now discuss about the structure of antibody. Glycoproteins, which are specific to specific antigens, are called as antibodies. In short, to fight against the antigens of different pathogens, antibodies are produced in the body, in the blood. So they are called as antibodies. So antibodies are nothing but glycoproteins which protect us from different pathogens or pathogens antigens. Immunoglobulins produced in response to antigenic stimulations, produced by plasma cells formed by B lymphocytes. Structure of antibody. It is Y-shaped molecule. There are two chains, that is two heavy chains and two light chains. Polypeptide chains are held by disulfide bonds. That means these chains are held together by bonds called as disulfide bonds. Hinge joins variable regions to constant region. Variable regions has antigen-antibody complex. That complex takes place in lock and key manner. We are going to study later on. And most of the antibodies are bivalent. So once again, just go through the structure of antibody. This topic is very important. Explain structure of antibody may come for 3 marks. So here is a diagram which shows Y-shaped structure of antibody. Here at the top there is a antigen binding site. So at this particular antibody, antigens are bonded. Then variable region of heavy chain, variable region of light chain, constant region of light chain. Constant region means the region which cannot be changed. Constant region of heavy chain. So down there is a heavy chain and the upper parts are light chain. You can see at the right side there is in cartoon antigen antibody complex is shown. Antigen antibody complex. combining sites of antigen is epitopes. You can see here where antibodies are joined to antigen. That site or that combining site is called as epitopes. Binding site of antibodies is called as paratopes. The site of antigen is called as epitope, while the binding site of antibodies is called as paratopes. So there is a combination of epitopes and paratopes. You can see in the diagram. You can see the epitope, that is a binding site of antigen, and below that there is a paratope, that is a binding site of antibodies. So here in this picture you can see lock and key arrangement or lock and key manner of antigen antibody complex. You just go through the diagram. Previous one, that is structure of antibody, antigen antibody complex. All these diagrams are very important. Let us now discuss about blood groups. Karl Landsteiner discovered ABO system of blood group. He found two antigens on surface of human RBCs, antigen A and antigen B. You can see there are 4 blood groups discovered by Karl Landsteiner, that is blood group A, B, AB and O. These are the 4 blood groups. You can see here in the nearby picture about the antigens A, antigen B. Then here you can see both the antigens, that is antigen A and B, and no antigens, that is blood group O, it shows no antigens. So in this chart you can see blood group AB, it has both the antigens, that is A and B, while there are no antibodies found. Blood group AB can be donated to blood group AB, can receive blood from AB, A, B and O. So this person who is having blood group AB is called as universal recipient because that person can accept or receive blood from any other blood groups, and therefore that person's blood group is called as universal recipient. While blood group A is having antigen A while antibodies B, it can be donated to blood group A and blood group AB. It can receive blood from A and O only. Blood group B is having antigen B while antibodies A. Just remember that antigen and antibodies are reverse. That means blood group AB is having both the antigens but no antibodies. Blood group A is having antigen A and antibody B, while blood group B is having antigen B and antibody A. Then blood group B can receive blood only from B and O. While blood group O, it does not have any of the antigen, so it is safe blood and it has both the antibodies, that is A and B, and therefore it can be donated to all the other 4 different types of blood groups, that is AB, A, B and O, because it has both the antibodies and no antigens are present, but it can receive blood only from O, and therefore this blood group is called as universal donor, as this person who is having blood group O can give or can donate his blood to any other person or any other blood group, and therefore called as universal donor. So AB is universal recipient and O is universal donor. Now you must have heard about Rh factor. Rh factor, it is discovered from rhesus monkey. Person having antigen D are called as Rh positive, while person without antigen D are called as Rh negative. You can see here in the nearby picture, the person who is having antigen D is Rh positive. Right side there is a Rh negative without antigen D. So the person who is having O negative blood group, this blood group is universal donor. That means it can be donated to any positive and negative blood group. Whether Rh factor is positive or negative, O negative person can donate his blood to any other blood group. Now when Rh negative mother conceives Rh positive fetus, destruction of RBCs of fetus takes place, that is called erythroblastosis fetalis. Now here the blood group of mother, if it is negative, and blood group of child is positive, that is if her husband is having positive blood group, there are chances the person or the child can be having different blood group. So that placenta is negative while that child or that fetus blood group is positive, and therefore there are some destruction of RBCs may take place. That condition is called as erythroblastosis fetalis. So in this case, immunoglobulin injections are given to the mother to change that placenta blood group temporarily. Okay, so antibodies or antigens are developed in mother's placenta to protect the child. That is called as to protect the child from erythroblastosis fetalis. So remaining topic of the lesson we are going to study in the next video. Thank you and have a nice day.